Hey everyone, today we're going to be talking about CRISPR and Cas9 technology and how it can be potentially a treatment for acute lymphoblastic leukemia and HIV. Feel free to meet our team, starting with Justin Johnson, Flora Brown, Steven Maradiaga, Wesley Collazo, Stephanie Ortega, and Kerwin Albaran. A 27-year-old man received diagnoses of HIV/AIDS and acute lymphoblastic leukemia on May 14th and May 30th, 2016, respectively. At diagnosis, the HIV viral load was 8.5 million copies per milliliter, and the CD4 cell count 528 million per liter. The infecting HIV was determined to be CCR5 tropic on the basis of previously described methods. The patient had undetectable plasma HIV RNA level and had lymphopenia. A 33-year-old male donor from the China Marrow Donor Program who had non-mutated CCR5 gene had a fully matched HLA type. On July 9, 2017, the patient underwent allogenic hematopoietic stem cell transplantation. Gene editing efficiency was evaluated by means of Sanger sequencing and deep sequencing of the genome of the recipient's bulk bone marrow cells, CD34 plus cells from the bone marrow, and CD4 positive cells from the peripheral blood. To investigate the potential off-target effects of the CRISPR gene editing system, whole genome sequencing was performed on edited cells after genome editing and after engraftment. Neutrophil and platelet engraftment occurred on days 13 and 27 respectively. After transplantation, lymphocyte counts and T lymphocyte subsets increased after transplantation. Acute lymphoblastic leukemia was in morphologic complete remission at week 4 after the transplantation. This remission continued over the 19-month follow-up period. In addition, minimal residual disease remained undetectable for leukemia-associated phenotypes on the basis of flow cytometry. The expression level of the Wilms tumor gene normalized to the Abelson gene, an increase in which predicts relapse, was less than 0.5% after transplantation, which was a level unchanged from before transplantation. So how exactly does CRISPR and Cas9 technology work? Think of the whole system like a construction site, where you have a contractor, a guy with a cement saw, a scaffold, and a building. The contractor represents the guide RNA, the guy with the saw represents the Cas9, the scaffold represents the RNA scaffold, and the building represents the DNA. So once the scaffold is mounted onto the building, the contractor is going to escort the guy with the saw within the scaffold onto a part of a building that needs to be modified. The guide RNA has his floor plans too, so he basically has a template on what needs to be done. The whole purpose could be for maintenance or an upgrade. The guy with the saw is going to cut a part of a building according to the template, so there'll be a chance to revitalize it. The same goes for CRISPR-Cas9 technology. Guide RNA leads Cas9 within the RNA scaffold while having a template of where to edit the DNA. Cas9 cuts the DNA strands into halves, so the sequence is open for modification. The cell is aware that the strands are cut in half, so it tries to revitalize the DNA sequence. CCR5 is the major infection site for HIV. Infusion of modified CD4 T cells at this site using the gene editing in previous clinical trials have shown to be effective in markedly decreasing the HIV viral load while reducing the decline in CCR5 modified cells. Other studies showed that using CRISPR-edited CCR5 ablated HSPCs in a patient with particularly HIV-1 infection and acute lymphoblastic leukemia tends to result in complete remission with full donor chimerism, with more than 19 months without gene editing related adverse effects. However, the percentage of CD4 positive T cells with the CCR5 disruption in lymphocytes was approximately 5%. So this indicates a need for further research into the CRISPR gene editing technique. So how exactly does CRISPR-Cas9 fare in HIV infection? It results in a successful allogenic transplantation and long-term engraftment of CRISPR-Cas9 edited CCR5 ablated HSPCs in a patient with HIV-1 infection and acute lymphoblastic leukemia. The donor cells engrafted with the full chimerism and the acute lymphoblastic leukemia was in complete remission for 19 months after transplantation. These results show proof of principle that transplantation and long-term engraftment of CRISPR-edited allogenic HSPCs can be achieved. However, the efficiency of the response was not adequate to achieve the target of curing HIV-1 infection. In particular, CD4 cells with CCR5 indels were continuously produced and received and released into the peripheral blood. It counts gradually recovering to the normal range in 6 months post-transplantation, while the patient was receiving treatment for the HIV-1 infection at the same time. This provides the patient with protection from opportunistic infection. In order to increase clinical safety, Cas9 ribonucleoprotein was introduced by means of non-viral transfection in order to avoid the introduction of exogenous DNA. So what are the ethics that center around this type of gene editing? Most of the ethical dilemmas relating to the genome editing center around human germline and how the changes made in the germline would be passed down to future generations with unknown genomic results. Researchers generally believe that human genome editing for reproductive purposes should not be attempted at this time. Most health professionals agree that it is important to have continuing public deliberation and debate to allow the public to decide whether or not the germline editing should be permissible. As of 2014, there were about 40 countries that discouraged or banned research on germline editing, including 15 nations in Western Europe because of ethical and safety concerns. In conclusion, the study described the long-term engraftment of CCR5 and CRISPR-edited CD34 cells after allogenic stem cell transplantation, which gave rise to less than 8% gene disruption in the genome of circulating bone marrow cells. The safety of CRISPR-based genome editing in the context of human gene therapy is largely unknown. CCR5 is a reasonable but not absolutely protective target for a cure of human immunodeficiency virus type 1. Because CCR5 no blood cells are largely resistant to HIV-1 entry.